Next slide, please. Good afternoon. I'm Mike Thompson. I'm with the Center for Urban Studies, and I'm here with Judy Westrick, who's from the Lumagen Instrument Center, and Ramona Rodriguez Washington, who's also from the Center for Urban Studies. And we're here to talk today about flooding in the Jefferson-Chalmers neighborhood and how that might be related to mold in the basements of houses that were flooded. This is part of the One Health program. Ours was a pilot project. And we also have, we're doing this as part of providing our stakeholders information about what went on in the study. Next slide. I'm going to go ahead and give a project overview in a minute, and then I will talk about flooding indicators and green infrastructure and some background and things like that. After that, Judy is going to talk about the types of molds that we found in the basement and the types of mycotoxins that were identified. Then we'll give everybody a chance to ask questions, and we'll see if we can construct useful answers. And then we'll move on from there, depending on how things go. Let's see, let's go to the next one, please. Next slide. Background. We're getting a lot more flooding in the Detroit metropolitan area and southeastern Michigan in general, and to some extent in Michigan. And this is really a result first of climate change. What is happening with climate change is that temperatures are going up. And as temperatures go up, the air can contain more moisture, more water vapor. So there's more water in the air more of the time. And so when there are big storms come through, there's more water that comes out of those clouds, and that can produce flooding. We also in Detroit have some very old infrastructure, sewage infrastructure, and we have combined sewers. That is, we put both, we use them for sanitary sewers and for flood purposes. And that means that they have a hard time meeting the capacity of these flooding conditions. And then the third thing is that we're dealing with housing stock that is, in many cases, over 100 years old. And so it's very, a lot of disrepair, and it's not, it wasn't built for these conditions. So what we're finding is more flooding among households in the Detroit community. We've done a survey of Detroit residents, and what we found is that over 60% of those surveyed have experienced flooding in recent years. That's both, we've seen multiple surveys to that effect. Let's go to the next slide. So what we wanted to do was to look at the consequences of this flooding with respect to mold. And we chose two target areas to look at. Jefferson-Chalmers, which was heavily flooded in the June and July events of 2021. It's 48215 as a zip code if you need to locate it. So that was sort of the area that was flooded, and we expected to see more mold in that area. And then the other area we chose was Southwest, which is on higher ground, much of it. And we saw less flooding in much of that area. There are parts of it that are low, but in much of the area, we saw less flooding. That's 48209. And our next slide, our hypothesis was that the flooded homes in Jefferson-Chalmers neighborhood, 48215, will have more mold and mycotoxins than houses that have not been flooded in the Southwest neighborhood. So we expected to see less mold in Southwest. we sampled in these houses in both areas, and we did a green infrastructure site survey in both neighborhoods. That is, we tried to analyze what green infrastructure was on each lot. Then the second thing we did was we did mold and mycotoxin analyses on the basement samples where we identified mold. And then we will be giving a written report of the results for particular houses to all the residents who participated in this, and give them strategies for remediation and so forth. Next slide. Some basic things that we found to begin with. We looked at 20 homes in Jefferson Chalmers 48215, and we completed a survey instrument there. We did 21 residents in Southwest. Then we identified what appeared to be mold in 14 houses in 48215, and found what appeared to be mold in only one sample in 48209. Wherever we saw what appeared to be mold, we collected it. And so we had these 15 samples. We did 20 site perimeter diagrams in Jefferson Chalmers and 21 in Southwest. And so next slide. And then I think you can just go on to the next slide, please. So we're interested in whether green infrastructure exists already in these houses. And if it does, is it soaking up water? And then we're interested in how water is getting into the home. And that will help us consider what green infrastructure we might put in place to reduce external flooding that comes from outside the home, as opposed to sewage backup. So let's go ahead and look at what one of our examinations of the homes look like. This is a little diagram we did for each of the perimeter observations. We tried to figure out whether there was a downspout and whether it extended outward, and where it didn't, we have this red blob on the blue downspout at the corners. That means that we didn't find a downspout there that was long enough to be useful. We also identified any trees that were next to the house. So you see one corner has a green tree next to the house. Then we also looked at the sloping of the ground next to the house. And so the two arrows that you see, green arrows, point towards the house. And those are two locations where we found the slope of the soil towards the house, which leads to pooling next to the foundation. And we also attempted to identify cracks in the foundation. You see one X on one of the foundation lines there. That was a crack we identified. And finally, we identified places where pooling had occurred. So the little blue elongated oval there on the left-hand side is an example of that. We also asked about any green infrastructure that they had. And then we surveyed the residents regarding whether they were flooded, and if they were flooded, where did the water come from? And did it come from outside the house, or did it come from sewage backflow? So that's one of the things we did. And I'll stop right there for a second before I move to the next slide and say that those surveys found that between 2/3 and 3/4 of the households said they had sewage backflow in the basement of the house. And the other 1/4 to 1/3, depending on how you get at it, had water coming into the basement from the outside. So many of the residents had both kinds of flooding taking place during one or more of the flood events in the summer of 2021. And so we now know that they're getting water in the basement from multiple sources. So let's go look at the next slide. We found that 15 of the 41 homes had mold. All but 2 were in Jefferson Chalmers. So Jefferson Chalmers had a high risk of mold, and there was a correlation of 0.637 between being a home in Jefferson Chalmers versus Southwest and having mold identified. So there were also positive correlations found between the lack of green infrastructure and basement flooding. Parameters with significant correlations were ponding, pooling time, the number of basement wall cracks, a negative exterior slope, meaning it sloped towards the foundation, and then lack of trees elsewhere in the yard. Over 40% of the people reported water above their knees when we asked them about the extent of flooding. And the extent of flooding was directly correlated with the presence of mold. If you look at this diagram, one of the external features of the house that is related to the extent of flooding is pooling time. What that refers to is the resident's assessment of how long the water pooled next to their foundation. And that was the variable that was the most closely related to the extent of flooding, and the extent of flooding was really closely related to whether mold was identified. That gives you a quick overview of the basic results of what our examination of the external house and all that, what that yielded. And so we can return to discussion of some of this later, but right now I'm going to turn it over to Judy, and she's going to talk about the molds that we identified and the mycotoxins that she identified. Judy.
So, hi, my name is Judy Westrick. I am the director of the Lumagen Instrument Center, and I'm going to discuss today 2 things: the mold that we discovered in the basements, and then what the mold, if it produced toxin or not. So the next slide is a quick summary on how we actually found the mold composition. So the people would go to their basement. If they saw a mold spot, they would scrape it off and put it into a 50 ml tube. They would scrape off mold for 2 tubes, and we requested to get as much as we could because we knew that sometimes the spots may not be big enough, and we needed a split sample. So we needed 1 sample to be tested for the mold test, and then we needed 1 sample for the mycotoxin test, which is the toxins that the mold produce. So the first test we're going to talk about is the fungal composition determination. So we used a test that we call ITS, or internal transcribed spacer. The idea here is we're going to extract the DNA from the sample. Then we're going to basically take that DNA and we're going to use what we call probes and primers. And we're going to look at a specific region in the DNA. That specific region, when we look at it, can tell us what genus of mold that we have. And then what we do is we call sequence, so we can amplify that so that we can make sure that we have enough data to see, and then we interpret that. So when we interpreted the mold composition data, next slide, please. Because it's just a sample from outside, and it isn't homogeneous, so it has multiple things in it, potentially multiple different types of mold. What we did is we identified it to what we call genus. So this test is actually only good to the genus level. And then you said, what's a genus? Well, if you remember from your biology class and you step down the classifications, you have kingdom, phylum, class, order, family, genus, species. We're at the genus level, so that's how we identified it. Something to think about is every genus has multiple species. Some of these species can produce mycotoxins, some of them don't. So all we know is that we have the genus present, and then we're going to identify that genus is potentially a toxin producer or not. So next slide, please. So here's the results. So we found that what we did is we took the top 3 from each of the houses and then counted them when we saw replicates of them. So Aspergillus is a mold, and we had 9 positive samples of them. Echinonium, we had 6, Cladospermium, 3, Microdinium, 2, and Penicillin, 2. And basically, these molds are a combination of food molds that most people will report in like food literature. They do have things, they do produce things that cause asthma, allergies. But let's get a little more specific now. So let's go to the next slide, please. So let's talk about the one that we saw most frequently, the Aspergillus. It was found again in 9 samples in the basement. It's been known to be allergenic and pathogenic, and actually also toxigenic, which means if it's allergenic, it's going to cause your immune system basically to have an allergy response. Pathogenic means it has a potential actually to grow inside of you. And then toxigenic means it actually produces the toxins. What we see in this is that it's very commonly found in damp places and water damaged places. And you can see pictures of it on the walls to the right to give you an indication of what it looks like. It's generally green, dark green, sometimes looks like brown hair, and generally also a velvet type texture. Next slide, please. So the next one is Acronymium. And again, you're going to see that it's pathogenic and toxinogenic. It's to the right here. And it gives you a good idea. This is a pure culture. And then you'll look up to the right of the pure culture and you'll see that you actually have, oftentimes you don't have a pure culture. So this will look like mold. And again, you'd scrape it off and we'd give you your results. And there's more than Acronymium present there. It's considered a household, again, mold, and it's commonly found in humidifiers, cooling coils, and dried wall that's been wet or insulation. It often has more of a pink, gray, orange color versus a green color. And its appearance evolves over time, and it's commonly might even look like, if you look in that dish, a little bit more like white dust versus a velvet. Next slide, please. Phantomspornium was found in 3 basements. It's mostly considered allergenic and not necessarily pathogenic or toxinogenic. It can cause, however, rashes and lesions. So from scratching, and again, that's more of an allergenic approach. And if you look, it's more olive green, brown, suede texture, and you can see the far right-hand side, it looks more suedey. And you can also see that it's also commonly found in things that are wet, such as wet, damp upholstery, carpets, floor cupboards. And then the next one, please. And the last one, no, we had 2 more. Marcodonium. This is an interesting one. There's not much known about it, yet we did identify it. It's commonly thought of as an agricultural mold. So you can see it's on the plants on the right-hand side. It looks like you basically powdered a plant, but we did see this in 2 samples. And I'm sure there'll be more that'll come out of this and more discovery over the next several years that'll give us some insight. And then finally, the last one that we saw was penicillium, which is the next slide. And penicillium does sound like penicillin. Not all things produce bad things, because penicillin, your antibiotic does come from penicillium. So, but on the it does produce other things such as allergenic, pathogenic, and toxinogenic compounds. It's actually of a higher risk to people that have a compromised immune system already. And again, it best grows in cool, damp places. It's also considered, one thing that we noticed is that many of these are considered food molds, which is a little different than what we actually see when we look at ocean studies and saltwater studies. They're usually more toward non-food molds, things that are actually just found in drywall and stuff. And then what does it look like? You get a picture of it there. If you notice, the other one was like white powder. This actually looks like it has rings going out. This is very common for penicillium. And you can see the darkness change. There's more color in this one. So you actually see a gray green, a white green, and also like an olive green in it. And then on the right-hand side, you can see some that's on the wall. Again, none of the stuff that's in the walls would be pure cultures. It's just the majority of it is that. So The next thing we're going to talk about is the mycotoxins. So these are the toxins that are produced by the molds that we found. And so basically there's every one of these mycotoxins has a class. So they all have what we call a common backbone structure, but they vary a little bit. And so you'll hear me talk about the class and the not the class, but they're all toxic and maybe just different degrees because they're backbone. So let's look at the identification and quantification technique. We use a technique called LC mass spec by mass spec, and that's how we're going to identify the mycotoxins. We're going to use what we call a targeted method. That means we have standards. So the standards themselves, whether or not we identify it. So we take our sample, we extract the mycotoxins, so we extract the inside of the mold, and then it's in a solution, and then we put it in this instrument. And if you look in the middle of this instrument to your right at where it says HPLC column, that separates the different kinds of toxins. And then it goes into the mass spec where it gives us what we call the mass of the compound. And then it breaks it apart, and then it's a puzzle. Then we put the puzzle pieces together, the different masses. So we're going to summarize it first, and then we're going to give you some examples of how we basically interpreted our results. So the next slide is our summary of our mycotoxins. And you look across the top, stegometicystin, patulin, satratoxin, and zearalenone is the 4 toxins that we identified. And if you notice, the stegometicystin is the one that we have a positive hit on it. You see an X. That meant that basically our mass and our pieces all matched the molecule you'll see below it. So that's the schematic drawing of the molecule. If you look at the patulin results, we had 4 patulin results, but you'll notice this is common to the family. And that means that basically we see the backbone, but we don't see the specific molecule. And again, you'll see that for the satratoxin and the zearalenone. Next one. So what does that mean? So next thing that we see here is that, okay, the stegometicystin is a carcinogenic compound. And it's basically produced by some of the mold that we saw. We saw the aspergillus and the penicillium. And it's going to target organs such as your liver and your kidney. And it's commonly, you'll see here again, they say it's going to be found in like coffee and foods, but we find it in our basements. On the right-hand side, you have a picture of the standard. And all I really want you to focus on is the 325 is the mass. And then you see the little pieces, the little lines going to the left that go to lower mass on the bottom. And then if you look at the one below it, that's our standard on the top. And the one below it gives you the exact same spectrum. That's why we know it's a hit. It's at the same time and the exact same spectrum. Then if we go to the next one here, which is patulin, where we identified it as not the exact, but in the family, the next slide. Nope, yep, there it is. The next slide, what you'll see is this is patulin. And what you'll notice is the standard on the top has a lot more little lines in the middle. But the big lines match. So the big lines imply that the backbone is the same backbone, and the little lines are saying they're not there. So we can't make a for sure identity of patulin. But patulin is toxic to just about to almost all living cells. Again, it's produced by aspergillus and penicillium, which we had in 9 of our houses and basically 2 of our houses for the penicillium. It can attack everything from your liver, kidney, lungs. So however you're exposed to it, it matters. So it can cause congestion in your lungs. And again, if you look at this, it's like wet places. But again, notice up on top front, it says mainly food, and then it's extracted down to, okay, you can also find it in wet places. So at this stage, I'm at the end of my talk, and we're going to take questions from the audience. Like, and I will look at questions. I don't know how to call on the person. So,
oh yeah, that's fine. I can just talk.
Okay, go ahead.
Yeah, I was just wondering, especially for people that are that live in these flood zones, do you know what they should do to prepare or just like make sure their house is okay during flood season?
So we went through the green, like, why don't you answer that? Because you actually have more background. I'm more of the chemist. I like to talk, but we're going to turn it over to you.
So there's two kinds of things that you need to do. One thing has to do with your sewer, and the other thing has to do with the perimeter of your house. If you're dealing with the perimeter of your house, in other words, flooding is coming from the outside of the house, there's water percolating up through the sewers, or there's just water running through the neighborhood and it's getting high, which is a lot of what has happened recently. Then what is really important is that, one, you've walked around the perimeter of your house and you've found any cracks that exist in your foundation, and you filled those cracks with some kind of silicone type gel that you put into that you shoot into those cracks. And then you turn around and you check on the inside of the basement and you do the same thing. The second thing you would do is to make sure that any areas where you have a negative slope towards the foundation, in other words, the dirt slopes towards the foundation, you would build up the dirt so that it has anywhere from a 10 to 35 degree slope away from the house, 3 feet or so. And in my case, in the back where I've had some problems, we built it up like that, and then we put plastic down before we put some dirt, and then we put the dirt down, and then we put some rocks on top so that it was hard for our friendly dog to dig it up. And so that's the sort of thing that you need to do. A rain barrel at any of the corners can be helpful. 6 to 8 foot extensions of the gutter away from the house. So it's taking the gutter way away from the foundation of the house, reduces the amount of water that the soil near the house has to absorb. That's important. Long term, having trees around the house reduces the amount of groundwater in your soil. Another thing that people can do, and fascinating, I was surprised to find this, but I found an article that shows that using those little aerator things that you push around the yard, has a concrete block on top and everything, it aerates the soil, that will increase water absorption as much as 20 to 25%. Also making sure that your soil is light, friable, to the extent that you can put more compost in it, the better. So those are some of the things that you can do around the house, plus a rain garden. And if you can flow one or more of those gutter extensions into the rain garden, then the rain garden can absorb a whole lot of water that would otherwise build up around the house. That's all out of the house. Inside the house, what we want to see is you to subscribe to the city program through DWSD that's gradually being rolled out to the areas that were flooded, that allows you to hire a plumber that the city will pay to put in backflow preventers. Backflow preventers prevents sewage from flowing backwards into the basement when the sewage system becomes saturated with flood water from up north down into your neighborhood. So that is a really important thing to do. A sump pump can help in that regard as well. But the other thing you should do before you put in the backflow preventer is to test your sewer lateral that comes out from the main sewer line that goes through the back of your property, usually along an alleyway in Detroit. And that lateral in Detroit, many of them are old, and if you put pressure on them, they can break, they can tear up. And so if you use a backflow preventer, the hydraulic pressure can tear up that lateral. So you may need to replace parts or all of that lateral if you're going to put in a backflow preventer. The backflow preventer can be put inside the house in the basement, or it can be put outside the house. In any case, what it does is it stops backflow into the basement. So those are the basic things that can be done, and that can make a lot of difference and save you from a lot of flooding in the basement. There's other things you can do, like putting your heater and your water heater up on blocks, putting anything on that's on the floor up on blocks. I missed one other thing about the outside of the house. We were out working on one of our pilot houses the other day, going around the perimeter of the house, getting ready to do the work that we're talking about. We found a window that was at ground level that was completely open. The glass was gone. I think there was a screen. That's not going to do you much good. And so what we're in the business of doing right now with that is that we're going to put in glass block window into that window so that it is completely closed off, so water can't rush into that basement. And this was at a house that was severely flooded before. Did I answer your question?
Yeah, thank you so much. It's just right next to us in my city, we have a flood zone, but they're trying to like make it into a new infrastructure. They're planning on building multiple houses on it. So I was just worried about like what would happen if the flood zone is gone.
Yes, you're smart to worry about that, because
Thank you.
you're putting structures where there used to be a place to capture water.
Mhm.
You need to attend your meetings and reach out to your politicians and tell them to make sure that the proper retention ponds are created to basically take care of that. And you should be reaching out. Which city or town?
Oh, I live in Dearborn Heights.
Dearborn Heights. Okay. So yeah, that would be the avenue that I would start on
Okay.
too, is making sure that everything, they have the proper retention they need outside of the areas. So
Oh, thank you so much.
Other questions?
There's a question in the comment
I
box.
got it. Yeah, it
Okay.
says, when gathering samples of mold, is it common to find 2 or more species when determining the species of samples? If so, how do you isolate? We don't isolate the species. So when we use a genetic technique, we can basically go in and determine, because we've done multiple versions of them, in this region, we look at all the regions and then find the differences of this piece in that region. So the actual piece is telling us which mold it is. And we can tell the difference of the pieces based on the type of instrumentation we use.
Other questions?
And we do separate the molds, the toxins themselves. We use a separation technique for the toxins.
Well, I think we have no more questions, and we've had a chance to talk this through. I appreciate everybody coming. Is there, if there's no further questions, we look forward to seeing you again at a future One Health, Healthy Urban Waters, Cures, or Clear presentation from Wayne State. And thank you for coming today.
Thank you. Have a good day.
Thank you. Ramona, do you want to stand for a second?
Stop the recording, please.
Yep.
Ramona, I didn't hear it say that the recording was stopped.